So welcome everyone, thank you for coming. My name is Thomasson and I am in the lab of Dr. Stephen Prue at UC Santa Barbara. And this is my talk entitled A Model for Coexistence and Divergence in Insect Vector Yeast Populations. So I might be slightly patronizing you guys by talking about what sympatric speciation is, but we all know what it is, but what we may not all know is that it's actually far more common, most likely, than most people believe it to, or at least believe it to be, or that it's originally been observed to be. And I think it's a really fascinating subject. I'm sure a lot of you do as well. It's understood really well in cases of things like allopolyploidy in plants and sexual selection and Margaritas. But there are a lot of factors that prevent its divergence from happening in these sympatric speciation events. One of the main ones is gene flow, but the one that I like to focus on is the concept of competitive exclusion within these interacting groups. So competitive exclusion is based on this idea of limiting similarity by MacArthur and Levins, and the idea there is that you have these overlapping niche spaces. The more they overlap, the more likely you're going to have heavy competitive interaction. And based on the axiom of inequality, if you have differences between individuals within a population, as in fact an incipient diverging population, or two separate species, as these things are usually described with, you're going to have differences, and therefore one is going to be better, and one will typically outcompete the other or competitively exclude the other. So if you imagine looking at, I think this works, yeah. So if you imagine looking at, this is your typical niche space diagram where you have resources and utilization. The more that these three different populations or species interact, the more you're going to have competitive pressure, and it's likely that the one that is best at utilizing the resource will eventually competitively exclude. So when you think about incipient species where they're actually almost identical in terms of their niche space, you would expect to see the highest level of competitive exclusion here. And so there is a huge field of study called coexistence theory that looks to evade all this competitive exclusion. And it's very well studied, as you probably know. But what's not very well studied is how this coexistence theory, how these ideas, these mechanisms within coexistence theory help to promote sympatric speciation by allowing the two populations to continue to coexist long enough to diverge. So one person that's looked at a type of coexistence mechanism is Tillman, and he has a model called the competition colonization trade-off model. And this is really the model that I'm basing a lot of my work off of. And how the model works is he imagined two competitors. He worked with grasses to start this model. And he had these two populations of different species working or living in the same environment, but one is considered the competitor. It's better at reproducing, it's better at using the materials, the resources available to it. And the other is not as good at competing, but really good at colonization. So it's able to disperse, it's able to get places faster, recruit new environments faster. And of course, when the competitor shows up in that environment, it will probably outcompete that colonizer, but it's this kind of patch space idea that allows these two things to, on average, coexist together and have about, have a consistent level, population level. And so as you can see here, you have this, oops, sorry. As you can see here, you have this competitor who has a standard kind of logistic growth curve looking effect, because really the colonizer has no effect on him or her, it. And you have this colonizer that does pretty well and then drops off eventually. But the idea here is that this was the first example of really stable global coexistence between two species that differed. And so I apply this to yeast because they have a really similar kind of competition colonization element to their life history traits. And so yeast are capable of both haploid and diploid reproduction, but in nature they tend to be constitutively diploid because in their diploid form they're capable of sporulating. And if you all remember from intro biology, sporulation is a very important aspect for yeast because it allows them to form a bit of a seed coating and be protected against the elements, desiccation, lack of nutrients. It basically allows them to shut off metabolically. So as a spore, you have this element of increased survival, but you're not able to reproductively increase your numbers, so your RS completely drops. So there's this trade-off. If you stay a vegetative state, you have this increased RS, you have lots of reproductive output, but you are more likely to die. And if you're a spore, you have to halt your reproductive success or your reproductive output, but you'll survive if something goes weird. So one thing that is particularly interesting about yeast is that they utilize insects for phoresis. They'll get ingested by all sorts of different insects and be dispersed all over the place. This is particularly the case in areas like grape vineyards where flies will visit to basically eat the rotting grapes and such. But they also pick up a lot of the yeast that are there, and then those insects will move them to new locations. And there's noted differential survival here. So Coluccio did a paper on this where she actually noted that if they were a spore, when ingested, the yeast were much more likely to survive than if they were a vegetative cell. So there's this trade-off going on with yeast, and this is really where my research lies. So in order to model the yeast system, it was really important to have resources involved in my growth curves. And unfortunately, there isn't really a direct variable of resource in a logistic growth curve. You have this K factor, which is indirectly resources, but not really resources. And because sporulation is triggered by loss of resources, I really needed to readjust my logistic growth curve, basically, and create a new curve or a new equation that would allow me to account for resources and not just rely on something like carrying capacity. So, a little bit of a crowded screen, but what's going on here is I have these two different, we'll call them lineages of yeast. We're going to call one population A, we'll call one population B. And the idea here is population A, which is here, has these two states. It has a vegetative state and it has a sporulated state, just like population B does. However, the difference between A and B is that A is going to persist in its vegetative state far past when the resources deplete at a certain level. It's referred to as the stubborn yeast, where even if there's barely anything left in the media that they're growing in, it still stays as a vegetative cell and eventually may go to a spore state, so it's a slow sporulator. And then we have the fast sporulating group, which gives up its ability to really reproduce a lot, but is more likely to survive if and when it is ingested by a fly, as most of my experiments are going. So instead of using K, I switched out K and started using a direct variable of resource. And to account for this, the fractional part of a logistic growth curve, I implemented Rmax as well in there, and then implemented an alpha factor for assimilation. And so it became painfully obvious after a few weeks of working with this that I understood why people just left it as K, but we'll see that in the next few slides here. So it did actually, the model actually did, or the equation actually did work in terms of getting what I wanted, which was a switching of the two populations in terms of coexistence. And so what you're looking at here is all 5 elements within this model. So that includes the fast lineage A in both vegetative and sporulated form, lineage B in both vegetative and sporulated form, resources, and I know there's a lot going on here, but I just wanted to give you guys an idea of how they looked all together, and we'll break it up. The most important thing to take away from this picture is that there is an oscillation happening, and there is the hints of coexistence happening, but that it's an unstable situation where you see this is actually continually growing, which is a problem, right? Because what's happened here is because there's no carrying capacity, we're basically kind of blowing through the ceiling, and there needs to be some sort of additional variable that is added in for frequency dependence of selection or some sort of resource like to draw it back a bit. So if I look at just the competitor strain, or the strain that's really good at growing and not so great at sporulating, we see a similar situation where there's a slight increase. The blue line is the vegetative, and the spore is the orange line here. And as you would imagine, as this one is increasing, it's using up resources, and because the sporulation is instigated by the drop of resources, we see that this one is also going to increase right around the time that this one is maxed out the resources. And so the processes are kind of behaving as they're expected. We expect these things to oscillate back and forth. The other thing I should note is that as part of the simulation, which is meant to model how a fly might pick up a collection of yeast and move it somewhere, we killed off a lot of yeast. We killed them off differentially, so a lot of the spores survived, a lot of the vegetative cells did not. But most importantly, the resources were replaced as if they were deposited, these yeast cells were deposited in a new location every 200 time steps. Okay, so that's why you're seeing this oscillation, is that really we were planting them in a new area that has, that's replete in resources. So the colonizer strain, very similar as well. It has that kind of oscillation element going on because of the resource replacement. But you see there's a lot more of a dramatic effect with its kind of out of control and unstable growth as we move through the time steps. So this is when we add together both the spore and the vegetative cell from each lineage. So this is really looking at population A in total and population B in total. And what you see is it's very weak, but there is a very similar oscillation going on here, similar to Tillman's colonization competition model. So we do see the trade-off there. It's just very weak and it needs a little bit of adjustment. And frequency, and we have to add that frequency dependency variable in. So how am I going to add the divergence factor to this? Well, there's a lot of different options, but Doubly and Deepman worked with using ecological character values. Osman worked with using genetic traits. And then the last thing that I hadn't told you is that there is this possible element of just looking at recombination in general, because when spores are transferred, they actually tend to digest into their haploid states, and so the rate of outcrossing increases in spores, whereas vegetative cells tend to stay diploid and there is less outcrossing, which may play a major factor. Additionally, these things will be fitted to a lot of the experiments and empirical work that's going on in the lab to give us better parameterization of the model. And with that, I will take any last questions if you have them.
So in the natural setting, do vegetative yeast maybe get transferred just like on the feet or something of the fly as well?
Yeah, we're looking at that. It's very possible that they do wallow around in the yeast and therefore there is some transfer, but the distance on that, we did a bit of a pilot experiment with dispersal. They don't tend to stay that long. So that's definitely something we could look at secondarily to see what the real effects are, but right now we're just kind of discovering whether the survivorship through the gut is what we expect.
Yeah.